Hello everyone, myself Liana Medhi. Let us read Fowl Cholera. Fowl cholera is a highly contagious disease of domestic and wild birds. Synonyms of this disease are avian cholera, avian pasteurellosis, avian hemorrhagic septicemia. Chronic form of this disease is known as wattle cholera. Etiology for this disease is Pasteurella maltosida, serotype A. It is a gram-negative, non-motile, non-spore-forming rod-shaped bacteria. The organism grows on meat infusion broth enriched with peptone or avium serum. Susceptible hosts for this disease are chicken, turkey, duck, geese. Other animals that are also affected are cattle, horse, pig, sheep, goat, etc. Mode of transmission for this disease are bird-to-bird contact, the oral, nasal, and fecal secretions shedding Pasteurella maltosida organisms. This requires very close and intimate contact with the affected birds. The next mode of transmission is ingestion of organisms. Ingestion of food and water contaminated by Pasteurella maltosida organisms. Consumption of infected birds by scavengers or predator birds. The next is aerosol way of transmission. Next, vectors as transmitters, which include mite, tick, flies, etc. The next mode of transmission is animal bite transmission. The next, transmission through fomites. Infection may spread through contaminated equipments, cages, hands of visitors, shoes, clothing, etc. Next is carrier transmission. Next, let us read about the pathogenesis. The organisms upon invasion may cause death of the birds within 6 to 12 hours, but the duration of death rate depends upon various factors like host factor. Those are age, sex, genetic difference, and immune status. The next factor is the invader factor, which are virulence of the bacteria, strains, dose, and the route of exposure. The virulency depends upon the capsule that surrounds the organism. Inability of virulent strain to produce the capsule may yield loss of virulence. Next is clinical signs. For the acute form, clinical signs are birds die after few days and reveal yellowish-green diarrhea with blood-stained feces, fever, cyanotic comb and wattle, depression, drowsiness and dyspnea. Discharges from beak and nose is seen. Death due to dehydration and septicemia. Usually broilers of 3 to 6 weeks get down on legs and die. Clinical signs for the chronic form are swollen and edematous wattles as seen in the picture. Joints may be swelled, which may lead to lameness. Mucoid discharge from the beak and nostril is seen. Conjunctivitis and pharyngitis may also be seen. Let us see how to diagnose this disease. Isolation and identification of organism with bipolar staining can be done for diagnosis. History and clinical signs like edematous and swollen wattles. Serological tests can be performed like rapid whole blood agglutination test, serum plate agglutination test, agar gel immunodiffusion test. Differential diagnosis is very much important to correctly diagnose a disease. So here let us see what diseases can be differentiated from fowl cholera. The first one is Pastorella gallinarum infection. This can be made based on culture and biochemical tests. Next, Ranichet disease, which is a viral disease, has a rapid course, high mortality, and no lesions in wattle and comb is seen. The next, fowl plague, which is also a viral disease. Extensive edema of head, neck, and wattle is seen here. Avian monocytosis. Here, dark blue color of comb, pale coloration of muscles are seen. Vitamin A deficiency. Changes in the esophageal mucosa, white pustules, and cheesy deposits, white deposits in the pericardium and kidneys are seen. For the treatment, sulfonamide like sulfaquinoxalin can be given in drinking water as 0.03 to 0.05% concentration respectively for 5 to 7 days. Broad spectrum antibiotics like gentamicin, chloramphenicol, and ciprofloxacin can be used. Penicillin can be parenterally used against sulfur-resistant flock. For prevention of this disease, the new chicks to be quarantined for few days before mixing with the old flock. Strict hygiene measures to be followed during an outbreak and generally as well. Try to separate the carrier birds from the healthy ones. Isolation of the infected birds are very important. Vaccination is available for this disease as commercial vaccine, which is the fowl cholera inactivated vaccine that is to be administered in 2 repeat doses to chicken of 8 weeks or older, which is to be repeated after 6 weeks. The doses are 0.55 cc, which is to be given by subcutaneous route. Thank you for listening. Have a good day.